So just to introduce our speaker this week, this is Edmond Rogers, who recently graduated his PhD at Purdue University in biomedical engineering. And before that, he did a master's at the University of North Texas, where he studied under Dr. Günter Gross. And that was where he developed the initial iteration of his platform for studying traumatic brain injury. So today he'll kind of show us, I guess, a lot about his PhD and all the progress he's made. Looking forward to it.
Thanks, Tyler. Yeah, the progress. A lot of stuff going to go through really quickly, but it's fun. It's really unique. Haven't really seen much like it. And yeah, hopefully we can have an interesting discussion about maybe some of the potential applications. But first of all, yeah, thanks, Tyler, for the introduction, the invitation. Thank you, ResearchHub guys. Tyler's been kind of letting me know a little bit about what's been going on. Sounds really interesting, super supportive. So thank you for hosting me. Again, my name's Edmond. This presentation is entitled Elucidating Pathological Correlations Between Traumatic Brain Injury and Alzheimer's Disease. So first, we'll discuss traumatic brain injuries, how they work and why they're important, followed by a brief overview of Alzheimer's disease. Then we'll detail the novel TBI-on-a-chip system and describe how we utilized it to conduct shared morphological and electrophysiological investigations into impact injuries and ultimately impact-induced neurodegeneration. Finally, we'll finish with a brief summary and some questions. Yeah, so I was just saying there's a lot of exciting new data that I kind of want to cover in this. It's about approximately 50-minute presentation, so I'm going to go pretty fast through the setup. But if you have any questions or want any more details about the background of the system, just ask me at the end. I have a ton of extra slides, maybe too many extra slides. So let's start off with TBIs, or traumatic brain injuries, which are the leading cause of death for people under the age of 44, and they create significant social and medical costs. The research has been complicated because the complexity of the central nervous system. It's a high number of variables that are just generally out of the control of investigators. The data is typically non-standardized. But of these traumatic brain injuries, mild TBIs are the most common, accounting for 85% of all the TBIs reported. A major problem with mild TBI is that the standard CT or MRI scans are not sensitive enough to observe this microscopic damage because there isn't any bleeding. And while significant efforts have been made to characterize these injuries, it's still extremely difficult to identify, yet alone linked to functional deficits. It's becoming clear that quantitative investigations are possibly only in vitro or in cellular models where tissue can be observed before and after injury, and cellular damage can be linked to functional or electrophysiological changes. So traumatic brain injuries can be characterized by two parts: the initial physical or primary injury that you're probably already familiar with and shown on the left, and then a delayed cascade of biochemical changes that are known as secondary injuries. Secondary injuries have already been demonstrated to disrupt cellular function through multiple mechanisms shown here on the right, including oxidative stress. Further, they provide the greatest opportunity for intervention strategies. Okay, so at the bottom here, you see a quick summary. First, you have the injury itself, which produces an immediate effect, but then you also have these secondary injuries that continue for the hours to weeks following that primary injury, which over the course of a lifetime can manifest as chronic neurological dysfunction, as we'll see on the next slide here. So here's a list of some of the negative and costly outcomes that have already been linked to primary and secondary injuries, which is just continuing to grow as we learn more. Today, however, I'm going to focus on Alzheimer's, which is a devastating neurodegenerative disease with no known cure. So as a brief summary, Alzheimer's disease eliminates cortical neurons and synapses, resulting in atrophy, and is the most common cause of dementia, which is a general term. It's pathologically characterized by the accumulation of amyloid plaques and tau tangles in the brain. However, our current diagnostic methods are only effective after the prime window for intervention has passed. So a link between trauma and Alzheimer's disease has been clinically established over the last 30 years or so. However, the underlying mechanisms are still not known. In response, I'd like to share a quote with you guys that I found in a Harvard review when I was just getting started with this idea. It is now clear that this complex pathology will require research on all levels, from holistic brain injury to cellular and even synaptic disruption. Which brings us to our aims for this project. So what if we could create a device that was capable of investigating trauma on the cellular level at both the short or immediate term and long term? Also, if we could separate primary from secondary injuries, explore relevant or clinically relevant comorbidities, examine TBI-induced neurodegeneration, and isolate force as a factor in protein aggregation. It would be great. That's what I started out wondering. And almost 9 years later, finally realized it. So this brings us to the development of the TBI-on-a-chip system. So first, we will very quickly review the methodology and its establishment in my first published paper before building a bridge to several neurodegenerative studies. Okay, so I know there's a lot of information on the slide, but if you could, please try and follow me here because it describes the methodology that will be used for the remainder of the presentation. So first, at the top left A, we see that E16 murine cortices, baby mice, are disassociated, pooled, and seeded onto custom microelectrode arrays in a histiotypical ratio, or a ratio of cells that is identical to the parent tissue. Our densities vary by the project, but we generally use somewhere around 50,000 cells per chip. The chips are optically transparent and great for microscopy, which you can see at the top right in B here. The phase contrast image shows a live network growing over about 6 of the 64 electrodes, 3 of which I have highlighted with yellow arrows there. The corresponding immunofluorescent images demonstrate the type of resolution available with the system and show neurons in green, microglia in red, and nuclei in blue. These chip-adhered networks are then secured in a proprietary miniature incubator or impact chamber and subjected to impact injury utilizing a pendulum design shown in C. We designed the striker arm of the pendulum to be variable so that both the contact time and G-force of the impact are totally adjustable. Also, the impact chamber and life support system summarized at the bottom left can maintain the network at 7.4 pH with 300 milliosmoles at 37 degrees Celsius for at least 1 week, in addition to tolerating up to almost 300 Gs. This longevity and stability is demonstrated in D, where I utilized extracellular recording electrophysiological capabilities of the system to compare one neuron's action potential profile over a 7-day period. During experiments, these profiles are logged for each neuron in the network as shown in the corresponding 2D raster plot and given timestamps for post-analysis of frequency and pattern extraction. But to quickly summarize and visualize large changes, we generally use the average activity profiles of networks, which is in terms of spikes per minute, or SPM, for the total number of active units or neurons. An example is shown at the bottom right in D there. To give you a better idea of what an actual chip looks like, you can see my gloved hand holding one there at the bottom of the center where it says Ed's hand. And while we fabricate our own MEAs in-house, we still utilize industry standard digitizing hardware and software analysis, which is Plexon at the bottom right-hand corner you can see there. Okay, so in our 2019 publication, we used the impact system and methods we just discussed to see what happens to network activity after multiple injury grades. Interestingly, an electrophysiological response profile to impact injury was revealed, which we describe as two-phase 3 plateau. At the top here, you can see a total of approximately 1 hour of activity separated by an impact, again with active channels in red and spikes per minute in green. So pre-impact reference activity is stable and shown to the left. Plateau 1 is characterized by 20% activity decrease at 5 minutes post-impact, and plateau 2 as a 10% decrease 20 minutes following the impact. Below, the time scales increase to 12 hours to show plateau 3, which is an average 15% activity deficit that occurs in the hours following the injury. It's important to note that even though the activity changes, the number of active units does not, which means that these changes are happening in the absence of cell death. In addition, a 20-hour control example with mock impact is shown as an insert at the bottom for comparison. But to summarize these experiments briefly, it turns out this profile was highly reproducible. So just to provide a quick overview, we found that the greater the force the networks experienced, the greater the resulting activity deficit. And that's demonstrated in this graph, which shows percent activity decreases against cumulative G-forces. Now, this was a really exciting observation for me, but I still had a few questions, like what happens after 24 hours? And are there impact-induced pattern changes in network communication? And how could we potentially recover these deficits? So we'll actually get back to all of these questions here in just a minute, but for now, let's examine the inquiry that I found most intriguing, which was, could this injury model be used to explore trauma-induced neurodegeneration at these ultra-acute time points? And so that's what we set out to do in our next publication. Use the TBI-on-a-chip system to examine impact-induced changes in oxidative stress through acrolein, a measure relevant to both trauma and neurodegeneration, as we'll see in just a moment. So first, we'll briefly review acrolein, followed by its spatial distribution and elevation post single and multiple impacts, and a possible mitigation strategy termed impact intervals. Last, we'll examine acrolein in the context of secondary injury and look at alcohol as a comorbidity. So acrolein is a reactive aldehyde and trauma biomarker that's already been linked to many neuropathologies, including TBI and Alzheimer's disease. In this cartoon, we can see that trauma initiates reactive oxygen species production, which is extremely short-lived and difficult to measure. However, this generates lipid peroxidation, creating acrolein, which easily binds the amino acid lysine, forming a relatively long-lasting target. Plus, acrolein has already been shown to damage multiple cellular components, which cause cell death and perpetuate the cycle, creating relatively greater neuronal damage when compared to other alpha-beta unsaturated aldehydes. Together, this makes acrolein an ideal tool for approximating overall aldehyde toxicity and oxidative stress, which we'll see on this next slide here. So first, we wanted to look at the elevation and spatial distribution of acrolein as a function of impact severity at 24 hours. And that's just a reminder, the same time frame as our 2019 electrophysiological study we just saw. Here we see acrolein as red, nuclei as blue, and neurons in green, with forces increasing from top to bottom, and separated as 0G or control, 30G, which would be considered a pretty mild injury, and then more severe injuries of 100 or 200 Gs. If you look closely, you can see more red appearing from top to bottom or going from A to D. And at the very bottom in numbers 1 through 5, I've highlighted some examples from the 200G merge where you can better see acrolein's distribution in neurons at pictures 1 through 3 and in glial cells in picture 4 and 5. Here, you can also see that the increases seem somewhat equally distributed between neuronal and non-neuronal or glial cells, suggesting that both participate in the pathological consequences, something we'll look at in a little bit more detail later. Further, most acrolein production is perinuclear, and while some appearance into the processes has been noted, it's much less prevalent. And this makes sense, as the nucleus is the most dense component of the cell, it will most likely have the greatest inertia post-impact, causing stress to the perinuclear membrane and generating oxidative stress, therefore more red or acrolein. So that morphological study paved the way for quantitative analysis using average intensity measurements for acrolein lysine adducts, which are shown as percent of control values for, again, single impacts of 0 or control, 30, 100, and 200 Gs. So on the left, we see that while mild 30G impacts only caused minor increases in acrolein, they were not statistically significant. However, if we keep moving right, we see that 100G impact significantly increased acrolein levels, as did the most severe 200G impact. In addition, when these average values are plotted as a function of acceleration on the right, the correlation becomes clear. But if greater forces increase acrolein production, could they also be cumulative? So next, we wanted to look at acrolein elevation as a function of multiple impacts using 5 and 10 rapid repeats of 30 and 200 Gs. For comparison, single impacts are included for both graphs. While I kind of expected to see these even greater increases suggesting compounding damage, it's really interesting to note that a single 30G impact circled on the left there, which again, reminder, by itself did not significantly raise acrolein levels, was able to surpass this threshold after multiple impacts. And that got us thinking, could we mitigate these acrolein elevations by simply increasing the time between injuries? Okay, so up until this point, we'd been rapidly administering impacts back to back with maybe 5 seconds between each hit. So now we're separating 3 mild 30G impacts by 2 hours each. So that's a total of a 6-hour period. And again, we see no difference between acrolein levels for a single 30G impact and control, which is expected. However, it significantly increases after 3 rapid impacts, which is now similar to what we saw on the last slide. And so after introducing this 2-hour impact interval, networks that experienced identical total impact forces of 3 30G impacts, just again, over a longer period of time, showed acrolein levels that were actually similar to control values, which is pretty awesome. I mean, if we could play with the force in time to clinically define recovery periods, it would be amazing. But this also got us thinking. Now that we're incorporating time into our impact protocol, how does this affect acrolein's role in secondary injury? Or could we even see secondary injury on this chip? So to both confirm acrolein's role in secondary injury and examine the model's potential for separating media-tied biochemical injuries from physical impacts, or secondary from primary injuries, we created what we call the conditioned media protocol, or just CM, with the acrolein scavenger hydrolysine. CM is the term we're going to use to describe media from maximally impacted cultures, or 10 rapidly administered 200G impacts 4 hours post-injury that is then substituted into non-impacted networks. So to reiterate, these CM cultures didn't experience any force directly, but were exposed to the media of cultures that did. We'll see this protocol a few more times, so just remember that CM relates to secondary injury. And to answer the question from that previous slide, yes, we actually can significantly raise acrolein levels without impact by exposing them to CM, which is shown here at the top right. What's even more interesting is that by removing acrolein from the media of the impacted cultures, using that hydrolysine acrolein scavenger, we were able to effectively cancel out these media-tied increases of oxidative stress, resulting into values that were very similar to controls. So in summary, while there are likely many trauma-induced processes at play, this evidence strongly suggests that acrolein performs a key role in secondary injuries, which we're going to explore even more in the next paper. Finally, there's studies that have shown alcohol consumption and the occurrence of TBIs are correlated. And unfortunately, the populations that are participating in these higher risk activities are also more likely to be consuming alcohol around the time of injury, which makes drinking a TBI comorbidity. So in the simplest terms possible, we wanted to try and mimic the experience of like playing in the big game and then going out to have a few beers afterwards to celebrate. Interestingly, an exposure of only 15 minutes to a concentration of alcohol, similar to mild intoxications in humans, was able to significantly increase acrolein levels at each corresponding injury level. What's even more interesting is remember that single 30G mild impact that didn't cause a significant increase in acrolein before? Well, as you can see in the red circle there, once you add alcohol, it does. This implies that alcohol exposure post-injury may be exacerbating damage by oxidative stress and likely other pathways as well. Okay, so now that we've established the system over the previous two publications and implicated acrolein as a key player, in the next manuscript, we wanted to kind of reach for the sky and continue building our bridge to neurodegeneration by refining our approach and simultaneously incorporating protein aggregation and inflammation into our investigations. Here, we'll follow a similar experimental progression, beginning with an overview of our markers. Side note, actually, this paper was just published as part of a special collection in Lab in a Chip journal, and we were in the running for the cover. We didn't get it, but it was just still really cool to even be considered. Beta amyloid ranges from 36 to 43 amino acids, but 40 and 42 are the most common. The pathological form is 42, which aggregates into the extracellular plaques that, along with the tau tangles that we'll discuss in a minute, define Alzheimer's disease. However, increases in intracellular beta amyloid 42 precede plaque formation and have been linked to both subsequent aggregation and even tau phosphorylation, among other mechanisms of interest. Yet few studies have made this direct link between trauma and beta amyloid 42. I especially want to mention Dr. Gurus' lab at Lund University, who works with intracellular beta amyloid 42, has been very helpful with these upcoming studies. So on the top right there, you can see an example of an extracellular plaque in brown. Note the size of the plaque at the scale of 50 microns. And if you look at the bottom right, you can see the relative size of intracellular beta amyloid 42 in black at the same scale. All right, so now you've kind of got an idea of what we're looking for. Let's look at it in our system. So similar to before, we first wanted to get an idea of what exactly we were looking for. Again, using green to show neurons and red to show acrolein, we now included beta amyloid 42 in blue and compared a non-impact control at the top there with an impacted network below. While impacted neurons and glia again showed heightened levels of acrolein, relative increases of beta amyloid 42 were also observed with some co-localization. You can see this better in the enlarged sections from the impact merge at the bottom, in neurons for picture 1 and non-neurons for picture 2. And while acrolein continues to present a somewhat diffuse pattern throughout cellular bodies and processes, beta amyloid 42 is much more punctate, with the majority of localization in neuronal cell bodies and some appearance in cellular processes, which I've highlighted here with the white arrows. Interestingly, beta amyloid 42 was also present in some non-neuronal cell bodies, just at decreased values relative to their neuronal counterparts. Okay, so that's what we're looking at. Now let's switch gears briefly and take a look at inflammation. Inflammation's already been linked to multiple neuropathologies, including Alzheimer's disease and TBI. In addition, it can be induced by acrolein and has been shown to further exacerbate beta amyloid 42 aggregation, which we'll discuss in just a few more slides. TNF-alpha, or tumor necrosis factor alpha, is a demonstrated biomarker of inflammation that is produced in response to trauma by primarily reactive microglia. And our system looks a little something like this. So here we kept neurons as green and acrolein as red, but changed blue to show TNF-alpha, which revealed very clear elevated perinuclear levels of both acrolein and TNF-alpha in the neurons and glia of impacted networks, with the majority localized in cell bodies and an interesting degree of co-localization. Again, at the bottom, you can better see their spatial distribution in enlarged sections from the impact merge for a neuron in picture 1, and again, a non-neuronal cell in picture 2. While you can also see some appearance in cellular processes, which again is denoted by the white arrows, it seems to be less common, with a minor preference for TNF-alpha. So looking at picture 1 and 2 side by side, the purple, or co-localized oxidative stress and inflammation, seems pretty clear. So again, we set out to quantify these changes along with the beta amyloid 42. Here we see that impact-induced elevations of acrolein were in the expected range in A and were accompanied by significant increases in both TNF-alpha and beta-amyloid 42 production in B and C. Further, if these changes are graphed with acrolein as a common variable in D and E, the correlation becomes clear. So if acrolein is correlated with impact-induced increases of inflammation of protein aggregation, what would happen if we could remove it? Here, for the first time, we tried scavenging impact-induced acrolein directly, adding hydralazine to cultures immediately following the injury. Interestingly, this approach wasn't able to totally eliminate these elevations, but it was able to significantly reduce not only the acrolein post-impact, but also TNF-alpha and beta-amyloid 42 production. You can see there in B and C. This strongly implies that acrolein is playing an important role in the impact-induced elevations that we just described. So then we wanted to ask the same question another way. If we can reduce beta-amyloid and TNF-alpha by simply removing acrolein, then is acrolein alone capable of reproducing these changes? And the answer is yes, partially it can. Here we simply replaced the impact injury in our protocol with a 4-hour dose of exogenous acrolein at a 50 micromolar concentration. And that's a physiologically relevant dose, which just means an amount that's been previously measured in patients. So as you can see, this exposure alone was enough to recreate about 50% of the impact-induced increases in TNF-alpha and beta-amyloid 42 that we just saw. So again, it's likely that acrolein isn't the only culprit here, but it definitely looks to be a major contributor. And if acrolein alone can cause these changes, and it's involved in the diffuse secondary injuries that we talked about in the last paper, then if we again utilize our conditioned media model of secondary injury, shouldn't we see similar changes? And as a matter of fact, we did. By adding media from the impacted cultures to non-impact control networks, we saw partial but significant intracellular increases in, again, acrolein, which fell in the expected range in A, and now TNF-alpha and beta-amyloid 42 in B and C. Maybe even more interesting is that when we added hydralazine, which again, that's our acrolein scavenger, we were able to almost entirely neutralize these increases, bringing levels back down to basically control values. Taken together, this again suggests acrolein's participation in secondary injury, and even more importantly, acrolein and secondary injury's involvement in generating inflammation and increasing protein aggregation, which are both two key contributors of Alzheimer's disease. And so we thought, if acrolein is playing such a major role in these mediatized secondary biochemical injuries, shouldn't we be able to see it in the media at relatively higher concentrations? And so we reran our conditioned media experiments, each with a special corresponding sham network that was age, batch, and procedurally matched, and analyzed the media content for relative levels of acrolein protein attacks using Western blot. The 5 trials are quantified in A and B, but as my PhD advisor used to say, seeing is believing. So I want to jump over to C and just show you how clear these differences were. This is an actual image from a gel from one trial, no cut or paste or crops, of pre and post sham in blue and impact in red. Again, we're seeing that these impacts increase acrolein, which propagates and diffuses into media, further damaging cells, even in the absence of impacts, like in our conditioned media model. So far, it looks like acrolein is involved in both primary and secondary injury and related to impact-induced inflammation and increases of beta-amyloid 42, which is exciting. But now I wanted to use the system to try and answer some more kind of abstract questions that have been rattling around my brain. This is one that I'm probably the most excited about. So quick review, beta-amyloid 42 is a pathological form of the amyloid protein. It is the most likely to form aggregates. But how and why? And what if both force and acrolein could directly alter these kinetics? So in a special set of experiments, similar to a method in a paper that I am on with Dr. Glenn Acosta, he's a former lab member, we purchased commercially available purified beta-amyloid 42, reconstituted it, and then split it into a sample between 3 different treatments: control, impact, and acrolein exposure. We analyzed the results at 24 hours later, and it was using STS page. So first, let's talk about impact. I found protocols that used prolonged physical agitation as a method to increase aggregation in beta-amyloid 42 solutions, but these were like very low forces, maybe just like a shaker, and it was usually over a period of hours. Then I learned that inputting mechanical energy, into the system was thought to be a way of expediting what would normally be a relatively slow nucleation step in the aggregation process. So I thought, what if impacts could do the same thing? They're much shorter in duration, but much higher in total force. And as you can see in A there, the difference is pretty subtle, but if you zoom into our area of interest in B, which is where the larger oligomers are hiding, and adjust the contrast, there's actually a pretty clear increase in the beta-amyloid aggregation for impacted samples, and that's highlighted by the red circle. Now, even more interestingly, is that when I was reviewing beta amyloid 42's structure, looking for clues about the impact aggregation process, I realized something crucial. Lysine residues make up about 10% of the amino acids in beta amyloid 42. Now, if you remember, that's the amino acid that acrolein most loves to interact with. So next, after searching the literature, I found a chemistry paper that described how lysine residues in beta amyloid 42 play a critical role in protein folding and subsequent aggregation. So we decided to test it. We added some exogenous acrolein into one of the solutions, and as you can see up there in B at the top right, this resulted in the most pronounced elevations of aggregation, and that's highlighted by the red square. Furthermore, now we're able to see that with this Alzheimer's disease-related protein, impact or acrolein exposure alone were enough to increase aggregation, even in the absence of a physiological system. Combined with our previous results that show endogenous intracellular increases of both acrolein and beta amyloid 42 and their modulation through exogenous acrolein addition and scavenging, it certainly seems like there's a strong connection. Unfortunately, I don't have as much time as I'd like to go through the last part of this paper with you, but just as a quick overview, I really wanted to incorporate my electrophysiological background to again confirm both our secondary injury model and acrolein's involvement. So quick refresher, this is just like the profiles we saw before, with spikes per minute in green and neurons in red for 3 separate, approximately 45-minute periods. Basically, we set up networks on a recording station and established their stable, native activity before blindly swapping their media with one of 2 treatments, conditioned media or media from other non-impacted control networks, to try and see if we could tell the difference just by analyzing the corresponding changes in network activity. A great example of a regular response to a normal media change is shown at the top in A here, where the reference activity is temporarily increased by the replacement of standard media from regular cells before returning back to reference activity. We can talk about this in more detail afterwards if anyone likes, but basically in this type of recording, this is the response that you would expect to see. Just a quick activity spike due to the relatively large physical agitation of the media against the cells, followed by more reference activity. However, if you look to the right there, every time that we added the conditioned media, or again, media from impacted cells, we saw the exact opposite, or an activity depression that stabilized at about 15% below the reference. Just a reminder, the stable red line means that our number of units isn't changing, which means that these functional changes are occurring in the absence of cell death, which is exactly what we'd hope to see. These control and conditioned media response profiles were thankfully obvious and relatively easy to identify. But when we scavenged the acrolein out of the conditioned media samples using hydralazine, it got a little bit more tricky. So they fell somewhere in between, as you can see at the bottom left there in B. But overall, acrolein scavenging minimized these deficits from an average 15 to 5%, which is summarized in C. Again, this data continues to show that while many trauma-induced processes are likely involved, acrolein seems to play a key role in secondary injury. Okay, so now we've looked at beta amyloid 42, which is one of the main suspects in Alzheimer's pathogenesis, and implicated acrolein as a likely key contributor. But what about tau, which was our other prime suspect? Can its dysregulation be triggered with an impact? And could acrolein also play a role in this process? How do both affect function? These are the main questions that we wanted to answer in this manuscript, currently in preparation and overviewed briefly on the slide, which is pretty similar to our previous experimental progressions. So first, a quick overview. Tau is a protein that is primarily found in the axons of neurons in the central nervous system and plays a part in cell signaling, development, maintaining microtubule stability, and a lot of other roles we're just finding out about. Neurofibrillary tangles, or NFTs, are aggregations of pathological forms of tau that have been hyperphosphorylated. This hyperphosphorylation compromises the protein's ability to bind and allows it to become displaced or mislocalized from primarily the axonal compartment to the somatodendritic compartment, and eventually into the extracellular space and ultimately NFTs. In other words, with tau, we want to do something similar to what we did before, using one of the final steps like the amyloid plaque, and then working backwards towards its contributing components. So we reached out to Dr. Lau at the U of M, who'd also been thinking that if mechanical injuries lead to Alzheimer's, then we should be able to see some sort of neurodegenerative processes initiated post-mechanical injury. And he actually, thankfully, described a method for using tau mislocalization to do just that. To give you some perspective, if you look at the top right there, you can see another section visualizing amyloid plaques, but now also with NFTs. But before these NFTs form, if we were able to look inside the affected neurons, hypothetically, we should be able to see more tau in the wrong parts of the cell, or just outside of the axon. But in order to get a 2-for-1 special and simultaneously obtain information about their activity, we decided to look at the dendritic spines specifically. Last summary note, dendritic spines are tiny protrusions from the dendrites that form functional contacts with neighboring axons. They're very plastic. They can change their shape, size, and density in response to neuronal activity. If you look at the bottom right there, you can see an example from Dr. Lau's group. That's where red represents tau and yellow both represents a dendrite segment and spines. The spines kind of look like small detached balls directly adjacent to the dendrites, which are denoted by white arrows. Okay, so that's what it looks like in a standard preparation. Now let's take a look at what they look like using our system. Pretty similar with some minor protocol changes that are briefly summarized in the top panel there. We're showing spines in green and tau in red for an impact and control network dendrite at 24 hours post-injury. If you look closely, you can see two key differences in the impacted dendrite at the bottom. That's an increase of yellowish orange spines, which indicates positive for tau and are denoted with the red arrow, and also an overall decrease in the total number of spines. I'll give you a count in a couple of slides, but for now, just focus on that bottom right enlargement of the impacted dendrite, which gives a pretty clear example of a tau negative and tau positive spine. So then we wondered, could acrolein be involved in tau's displacement? And using this more resolved method, could we see it in the spines? Interestingly, we couldn't find anyone who'd ever tried to visualize oxidative stress in spines. This is actually a first. The results are certainly more subtle than the tau was, but acrolein is definitely there and has an increased appearance in impacted dendritic spines compared to control. Again, I've isolated a pretty clear example down there at the bottom right, where you can see an acrolein positive versus negative spine. But yeah, it's pretty clear. So this generated a lot of questions about how tau and acrolein are related, which we'll address in just a minute. But for now, let's quickly put a number on these morphological observations I just sped through. So using Dr. Lau's method again, we wanted to compare 3 parameters going from left to right for control, which are blue, and impacted, shown in red networks. The average number of spines present and whether or not they showed positive for tau or acrolein. Interestingly, impacted dendrites had almost half the number of spines when examined at 24 hours post-injury, which implies major changes in cell-to-cell connectivity, maybe even similar to what we saw back in the electrophysiological profiles shown in our 2019 publication. In addition, we also saw that of the spines that remained after the impact, twice as many were positive for tau and 3 times as many were positive for acrolein, both of which have significant functional implications. So again, we see a correlation and we're curious, is acrolein alone capable of recapitulating these impact-induced changes in both total spines and tau mislocalization? And the answer was a resounding yes. Acrolein exposure alone was able to reduce the total number of spines, again, by roughly 50%, as you can see in A there, and also dramatically increase the mislocalization of tau into dendritic spines, which is shown in B. So now this really got me thinking, how exactly is acrolein able to displace tau from the axon into dendritic spines? So over to the right, I took a closer look at what exactly connects tau to the microtubule in the axon, and guess what? The area on tau is referred to as the microtubule binding region, or MTBR, and contains several lysine residues, which recent studies show are key to maintaining this connection. If you recall, lysine is the amino acid that acrolein reacts most readily with, so it's quite possible that this increased acrolein generation, whether it be through exogenous addition or impact, is disrupting tau's ability to bind, which over the next 24 hours enables its diffusion out of the axon and into the rest of the cell. And as interesting as this mechanism is, I want to leave it behind for just a moment and get back to our story. So more or less, we're now also demonstrating an in vitro model of taupathy, more specifically trying to link mechanical deformations with abnormalities in tau proteins, in addition to examining acrolein's role. And while to this point, we've been describing impacts as uniform G-forces, which are an accepted measure, we thought, wouldn't it be cool if we could really try and better understand the forces acting upon the cells more directly? And so in collaboration, I had to throw that in there, Tyler, with the very talented Ray's laboratory, we were able to construct a digital twin of the TBI-on-a-chip system and use a finite element model to predict shear stress through the MEA during impact. And you can see a fast summary of the results there at the bottom right of the screen. To be honest, there's a lot of information here that could probably be better explained by Tyler. Again, thanks for hosting me. But to summarize briefly, first, we performed 2 different G-force simulations, 30 and 200, or R minimums and maximums. Next, we used those results to identify the 2 areas with the largest differences in stress. One area is termed center, and that's our standard seating location. The other new location is called edge, which are both labeled there in B. So then we averaged the total amount of shear stress experienced by those areas, or time averaged shear stress, for the 5 milliseconds immediately following the impact, and compared the two areas side by side. And you can see it in C there. Interestingly, using this measure, both G-forces resulted in significantly different shear stresses between their respective center and edge locations. However, the almost 10-fold difference between center and edge locations in the 200G impact gave us another idea. What if we could experimentally verify this model using the same morphological measures we just saw, but by using two separate networks grown in the same plate at these new center and edge locations? Oh, yeah, this was a tough one. It required a lot of changes. I normally don't talk about this part, but yeah, Tyler and I really struggled through this portion. So it's a dual network approach that we'll see. But yeah, the results were absolutely fascinating. Here we use those results from the digital twin to spatially guide our initial culture seeding during network generation, producing what we call a dual network technique, which again features one standard centralized network, termed center, and then one network that's off-centered called the edge location that we just discussed, and again was predicted to experience significantly greater stresses. So we use the same methods to detect and quantify the total density of dendritic spines, in addition to the presence of tau and acrolein, from neurons at both the center and edge locations in the control and impacted networks. As expected and shown at the top there in A, B, and C, no significant difference was shown between center and edge locations in control networks for any treatment. That made sense. So then next, we looked at D down there at the bottom left, and we could see that neurons from the impact edge networks revealed a minor but significant decrease in average dendritic spine densities when compared to their impact center networks. And of those spines in the impact edge networks, there was a significant average increase in the presence of both tau and acrolein, and that's shown in E and F. Oof, as briefly as possible. Yeah, there's a lot of information on this slide too, but we wanted to test network responses to a 30G impact and 200G impact and acrolein exposure, which in terms of this graph move in columns from left to right, comparing minute average activity for the 5 minutes immediately before and after each treatment, looking for changes in not only the spikes per minute or SPM like we usually do, and that is shown, which is shown in summarized in the top row A there, but also now changes in network dynamics using average burst rate and average burst amplitude shown in rows B and C. Now, there's a whole other full-page figure that goes with these graphs, summarizing individual unit network response profiles, but for the sake of time, I'll just tell you that they matched our 2019 publication. In addition, here again, we see significant changes in spikes per minute for impacts of 30 and 200 Gs. But now we also see that acrolein exposure alone can cause deficits that seem to fall somewhere in between the two. Now, the next two measures, B and C, of burst rate and amplitude, require a fairly extensive explanation, and we can discuss it in more detail after the presentation if anyone's interested, but quite literally, they describe changes in the firing hierarchy of the neurons that make up these networks. This means that not only is there a change in firing frequency, but higher G-force impacts and direct exposures to acrolein are capable of altering the patterns of connectivity that these networks produce. This both confirms the hypothesis presented by our 2019 publication and gives us some interesting new information about the time scale at which these impact-induced alterations occur. So my hypothesis is that the impact-induced increases of acrolein that are generated in the milliseconds following impact immediately disrupt tau binding in the axon and also cause membrane damage that alters spine morphology, which is a reminder, extremely plastic and responsive to neuronal injury. And in addition to inflammation and aggregation, all of this together ultimately affect network activity. And while many trauma-induced processes are probably contributing, acrolein likely plays a key role, since many of the same pattern changes can be recreated through acrolein addition alone, among the other reasons that we've just demonstrated. And while I was fascinated with these neuronal functional changes, I started to wonder, was there a way that we could look at functional changes for any other cell type in our histiotypical culture? Which brings me to our final section. Here, in collaboration with the Chopra Group for a paper that's currently in preparation, we decided to also investigate corresponding changes in microglia function post-impact, with a few additional exciting changes that we'll see here in just a second. So again, following the same general pattern as before. So microglia are the chief immune cell of the central nervous system and play critical roles in maintaining overall brain health, which includes scavenging for plaques in NFTs using phagocytosis, which you can see on the right there. While we still have a lot to learn about microglia, to summarize quickly, they are currently classified as being in one of two major states, depending on the situation, ramified or reactive. However, somewhere in between these two extremes exists a middle ground, where their phagocytic capabilities are impaired, and it's already been linked to both inflammation and TBI. In addition, current studies suggest that over extended periods of time, these impaired states could be connected with Alzheimer's disease pathogenesis. So in response, through some minor modifications to the impact system and protocol, we developed a method for evaluating both microglia and neuronal function. Now it's 7 days post-impact. In order to evaluate microglia function, we needed a new approach. Again, for those non-neurophysiologists out there, the microglia are not electrophysiologically active as the neurons, so we had to modify the approach simply. And since we're familiar with the well-established beta amyloid 42 that we've already confirmed its relevance here, plus it's been linked to changes in neuronal activity, as we'll see in just a moment, we decided to use it to also evaluate the phagocytic capabilities of microglia at 7 days post-impact. So using a compound called PHA-beta, a human beta amyloid 42 analog, which exhibits green fluorescence after internalization in acidic organelles, but not at physiological pH. We found that the happy control microglia, shown in the red at the top left there, readily phagocytosed the exogenous beta amyloid without a problem, which then put off those scattered little punctate yellowish green signals, and just as expected. But interestingly, if you look at the impacted microglia below that example, the reduction in uptake, or yellow dots, is quite clear, even at 7 days post-impact. And when we quantified those changes, there was about a 50% reduction on average. In addition, when we looked at their neuronal counterparts in the top right there in blue, we actually saw a slight but significant increase in the intraneuronal appearance of exogenous beta amyloid post-impact. Now, this makes sense because the more beta amyloid should be available in the media if the microglia can't remove it, which also has some interesting functional implications. And because acrolein is known to cause membrane damage, and we've already shown that it's increased post-impact and capable of propagation, that if it's still elevated at day 7, it could also help facilitate the entry of PHA-beta into the neurons. So we decided to check. Interestingly, acrolein was still elevated by approximately 10% at 7 days after the impact, with similar subcellular spatial distribution in both neuron and glia cells, including microglia. And that's what we covered earlier in our 2022 paper. Again, in context, this makes sense, as acrolein diffuses and generates more acrolein through lipid peroxidation. So then I started wondering, if acrolein is still elevated to day 7, it has been shown to cause deficits in network activity, then could we still see those deficits at day 7 after the impact? Excuse me. And interestingly enough, we did see activity reductions still present at day 7. And in fact, they were similar to what we saw at 24 hours in our 2019 paper. More specifically, by comparing 20-minute segments of average activity in sham and impact networks at day 1 before the impact with identical segments at 7 days later, we found significant deficits in network SPM that were distinctly absent from sham cultures. Further, when we went to add our non-toxic dose of PHA-beta to evaluate microglia function, we also found an increased neuronal sensitivity to exogenous A-beta, which produced an additional approximate 10% deficit in activity that is shown on the right. As expected, there's no change in the procedurally and age-matched controls for any of the treatments. Finally, we thought, yeah, so if acrolein is truly playing a part in trauma by propagating cellular damage that can still be seen 7 days later, then shouldn't we be able to recreate these deficits, at least partially, through the exogenous addition of acrolein alone, even in the absence of impact? And sure enough, acrolein addition alone was capable of partially but significantly reproducing the impact-induced reductions in both network activity, shown on the left there, and microglia phagocytic capabilities, shown on the right. And that's all at 7 days post-injury now. So this again suggests that while many trauma-induced processes are likely at play, acrolein could play an important role and might even represent a viable therapeutic target, which we're exploring more now. So in summary, this new device, TBI-on-a-chip, is capable of generating graded, consistent traumatic injuries, can provide real-time morphological and electrophysiological access to cells simultaneously, can be used to separate primary and secondary injury, easily used to investigate clinically relevant comorbidities such as alcohol, we talked about, can serve as a semi-high throughput platform for testing potential pharmaceutical interventions like hydrolysine, and even recovery periods and mechanisms. Remember the impact intervals we talked about? Can also reduce the use of experimental animals required. Now, I'm not saying replace, but it can definitely reduce. and it can be used to investigate the mechanisms that link trauma and neurodegeneration due to the unprecedented resolution it offers with regards to trauma. Now, these studies specifically suggest that oxidative stress, inflammation, and acute aggregation are connected, and that acrolein could be a key player. So we saw that inflammation, oxidative stress, and aggregation were all increased post-injury. We saw that mechanical force alone was capable of increasing aggregation in our cell-free trials. We also saw exogenous acrolein exposure, increased inflammation, aggregation, thalamus localization, and produced long-lasting deficits in microglia neuronal function, and that some of these changes could even be mitigated via acrolein scavenging. So therefore, it's possible that an initial impact-induced acrolein surge via membrane damage instigates an inflammatory response, which impairs microglia phagocytosis, thus inhibiting the homeostatic removal of pathogenic forms of amyloid and tau proteins, which, as we just described, are now elevated post-injury. These elevations of pathological substrate accelerate aggregate formation, further exacerbating oxidative stress and inflammation in the system, creating a vicious cycle of damage. In addition, we showed that mechanical injury alone is capable of priming pre-existing beta amyloid 42 towards aggregation. This, combined with the now increased levels of beta amyloid 42 brought upon by impact, means an even greater likelihood of aggregation. And the fact that we've already shown both acrolein to be elevated post-impact demonstrated its ability to both increase beta amyloid 42 aggregation in the absence of cells and recreate multiple characteristics of injury in our model, and even reduced many of these impact-induced changes through acrolein scavenging, continues to suggest that acrolein plays a key role in both primary and secondary injury. A finding that's also been echoed by our lab using multiple models of animal trauma. Finally, we've shown that impact injuries result in reproducible and significant alterations in both spike production rates and network connectivity, resulting in response profiles that can even be seen after mild injuries of 30 Gs, an impact force which did not raise acrolein levels significantly. Taken together, this information could be used to guide new non-invasive electrophysiological-based diagnostic methods for identifying even mild brain injury in patients, replacing current criteria which are largely based on patient feedback. Currently, we're moving forward with both acrolein scavengers and membrane repair agents as potential intervention strategies, and translating these results to in vivo models of injury, including recording, in addition to utilizing the model to investigate different injury types and their associated risk factors, while also incorporating changes into the system, like a new ALDH2 deficient mouse model. Then that's the enzyme that oxidizes reactive aldehydes like acrolein. We're working with human stem cells and even 3D cell cultures. So a lot of really exciting stuff is happening now. But I'm probably most excited about an upcoming collaborative project over the summer where we're trying to model cell skull dynamics during blast injury. And on one final note, I'd like to take this last 20 seconds or so to give you a quick virtual tool of the TBI-on-a-chip system, which for fun features perhaps the first ever classical guitar duet with an MRI machine. I like the Tyler Ed production. And on that note, let's see. I'd like to thank all you so much for your time, everyone in our lab and our collaborators who helped make this project a reality, and everyone at Purdue BME. Thank you guys. On that note, we'll take any questions.
Ed, that was awesome. Great presentation. I need to send you the updated video for the the duet there where it actually explodes and you can see the inside. So I'm glad you put at least one.
I've got the new music ready too. It's awesome.
Nice,
it sounds a lot
nice.
better than me choking. So
Just no worries. Yeah, I have a ton of questions, but does anyone else have one to start off?
I've got a boatload of questions. So maybe we can alternate time. Maybe we can alternate because I don't want to take up all the time. But yeah, Edmond, that was an awesome presentation. I mean, I was going to ask you about the glia cells, but you got to it at the end already. So that was really awesome to see. And I may have missed this at the beginning, but what kind of cultures are you using here? Are these primary cultures, primary mouse cultures, or are these like iPSC derived? What are they?
all this work. Also, thank you so much for that comment. Like the glial cell ignorance it's a problem in neuroscience research, and I'm guilty of it too, right? Like at the last step, I was like, we've got to do something else. I'm so tired of just looking at this. We have I worked so hard to get all these cells to grow, not just the neurons, which is what most people do.
Mm-hmm.
And yeah, anyway, thank you so much for saying that. And yeah, these are primary mouse cultures. They're embryonic generated, so like E16. And yeah, we are getting now to iPSC and some other models as well, but all that data is from primary neurons.
Primary neurons. Any particular region of the brain? Is this cortical or hippocampal or all of the above?
Frontal cortex.
Frontal cortex. Okay. So, and you said mouse, not rat, right?
Correct.
Mouse. MCCs. Okay. Got it. Maybe I'll pass it over to Tyler before. Actually, one last one before is what are you using to stain the acrolein? Because I'm doing some co-culture studies right now, and I need to look at some kind of like cell death apoptotic type marker, and I'm thinking acrolein might be a good one here.
Well, first to answer your question, it's the 2H2 by Stressmark. Our publication is their example image now, which I'm super proud of. But yeah, it's this little Canadian company called Stressmark, like MARQ rather than a K. Don't know why. Great stain. If you're looking for, is it live dead or you just want to show stress?
I just want to show stress. I'm doing conditioned media as well, and essentially treating iPSC-derived microglia, doing conditioned media onto iPSC-derived co-cultures of neurons and astrocytes. And I want to show that the neurons are getting wrecked, for lack of a better word.
Yeah, then I strongly recommend it. Also, the TNF alpha was like incredibly idiot proof. When I was working with the acrolein took forever to I spent a year dialing that in. It was kind of a pain. Tyler can share my information if you want to reach out. I can share like whatever I've got protocol wise. But like, yeah, the TNF alpha is a more readily accepted marker of inflammation, the kind of like a damage of sort. It is so easy to use. I can send you that brand too if you want, whatever we used. But yeah, just if you're open to suggestions, of course I'm pro acrolein now because of the lab that I worked in. But also, man, that TNF alpha, it's like really I keep saying idiot proof. I don't know. I used it just fine, so I felt like it's pretty easy to use.
Right on. Yeah, I'll get your info from Tyler after this for sure.
Sure.
Sweet. Yeah, I'll throw it out there. So Ed, for the 3D cultures, how are you guys planning on impacting them? Is it the same thing with the pendulum, or do you want to do something where you actually activate stress like uniformly through the whole tissue?
You know, Tyler, that's an excellent question. No, it's like I just don't want to talk too much.
Makes sense. Yeah, we can leave it at that, and I could pivot if you want.
Oh, no, it's fine. I mean, as in I'll just keep talking, not as in like I'm afraid to share or something. Okay. So that's kind of what I'm talking about, us pivoting to like the different injury model, because there's not a good way to impact a 3D culture. If you can kind of see my screen, you've got the glass that is growing. You just imagine a plate of glass and you're growing a brain on top of it. When it's just one cell at a time, works perfectly because the forces, they're divided uniformly across the plate. They all experience, well, as we saw, slightly different, but pretty much the same. When you start putting a ball of cells on top of it, like the top moves different, creates a different kind of shear stress. And like our preliminary experiments went okay. They didn't fly off the plate, which is what I thought was going to happen. But we wanted a better way to, a better injury model. So that's why we started pivoting towards the blast. The 3D models have been slowed down for the impact because of those problems I just said. But I think they're really going to come to fruition, or maybe maturation, I should say, like with this new blast model we've got working on. So, and again, just to give you a brief, similar description visually, you got this plate of glass. Now we have a ball of cells. Let me flip it upside down. Sorry. Ball of cells on top. If you blast it just straight down, it's a pretty even approach. I didn't answer that very well because that creates a lot more problems. That's why we're blasting from the bottom, so you get an even distribution through the glass. But anyway, we have thought about these problems.
Yeah, I kind of figured you'd say blast. I only mentioned it because when you were presenting, I just thought about like you could do a rotational injury where like say you're spinning it around a pendulum, and then all of a sudden you halt it, and everything kind of slides to a stop against the wall, the reservoir. It might be easier to model, is why I say that, just kind of selfishly.
That's funny. Yeah, that's really cool you said that. I didn't realize that. But yeah, you're absolutely right. Even with Jerusalem, the Oxford professor, yeah, like that's what he was kind of going towards, those directions of like, yeah, modifying the actual acceleration forces on the plate. Yeah, I just ran out of time. I spent so much time trying to like build the model that we had. By the time that it got to all the cool stuff that I saw that could have been done with it, I mean, we just finally got the first one accepted, so we could start using it. So
yeah, this is definitely like V2, V3 for sure, but that totally answers it. If you want to take one, or anybody else.
Yeah, if anyone else wants to go, if you should jump in. Yeah, I will monopolize this if I was left to my own devices. Okay, I'll hop in with one more. I was wondering, because this is all like so overlapping with my research that this is really great to see. I guess if you guys have looked in any of the in vivo models, I guess one of the things that gets lost during some of the in vitro stuff is any like peripheral cell recruitment into those areas, right? And so in TBI, you have some like chemotactic cues that pop up, especially in certain regions of the injury. So do you guys see that even when I mean, I guess, have you seen it in an in vivo model where if you do this acrolein scavenger, do you still see kind of the similar effects that you're seeing here in the in vitro system? If you have any prelim data on that?
Yeah, are you asking like, is it also like the spatial, maybe some of the spatial considerations of an in vivo approach? Like, because we do have like control cortical impact and like a few other models that we've been trying. Is that what you're?
Yeah, I was mostly wondering if you see similar things because like so if you have like a bunch of like peripheral inflammatory cells being recruited into that area, is the acrolein scavenging sufficient enough to hamper a lot of the damage you're seeing, even with because like you're right, microglia and astrocytes are like some limited number in the brain that are say secreting some kind of inflammatory products, but when you add in some peripheral cells that you don't see in vitro but in vivo you would, those peripheral cells being recruited because a lot of chemotactic cues pop up in the brain, is the acrolein scavenging still enough to like say, yo, we're all right?
Yeah, sorry, I misunderstood you at first. Yeah, it's like each answer that I give you is a few steps away. So like our impact model in vivo is still being published, but the in vivo blast model, hydralazine was able to actually improve the results quite a bit. So like it's all slightly different. And I assume it would be the same, but there are subtle differences between the two, right? So like, yeah, hydralazine has worked. Someone is looking specifically at that, but it's in the other group. It's one of the other groups that I was talking about working with. So like, I actually am not sure at this point. But yeah, the SHE side of the laboratory did show some preliminary, very positive data for hydralazine in animal trauma models.
Oh, that's awesome. That's really cool.
Yeah, and I don't even think I mentioned it, but it's just a repurposed. It's already been FDA approved and everything, right? It's like it's a drug that's just already out there, but it can be repurposed. So yeah, if we can get some good out of it, that would also be very good.
Those are the best ones. You don't have to go through a entirely new like adverse effects profile to figure it out.
Yeah, no, I definitely can't afford that. So
fair enough.
I'll throw another one out there. This one's a little harder. Ed, do you think it's like eventually of value to implement some sort of like fluid washing system? Because natively in the brain, you have the lymphatic system, which kind of passively takes away some of these solutes very slowly over time. So I was wondering, like, do you think it'd be relevant or even worthwhile to check how that rate actually affects the accumulation of acrolein and then also like the return to signal normality?
Yes, I would. That's all right. Let's see if I can get this a little bit higher. So in the life support system here at the bottom left, I don't know if you can see that. Yeah, there is a peristalsis like included in the system. And unfortunately, like, yeah, we can't, it's meant to supplement animal models, never replace. So like that filtration is something that would be very difficult to design and build for both an engineer and a physiologist. Like, but it's possible, but I just never know how accurate it would be my fear that you're selling people something that's not what it says to be. The closest that we came to was this, like, my advisor used to say, this made me laugh so hard. The solution to pollution is dilution. And so oftentimes we use, not in most experiments, there's like a large media reservoir that is slowly pushed through the system at 0.2 mLs per minute, I believe. Don't quote me on that. It's in the paper. And it slowly provides that type of movement that you're describing. It's lacking the filtration system, but the idea is that the media reservoir volume is so large and the experiments are so short relatively that it never actually becomes an issue in our setup. But I do like that.
Okay.
That's
No, that actually makes a lot of sense. And I forgot about the peristalsis in this, believe it or not. And I think the rates they're not too far off. And it's not well characterized in vivo yet of actually how much the flow rate is across the brain tissue from what I've seen. So yeah, it makes total sense.
We have a whole different series of experiments. They're never published, just about how flow rate affects activity, which is actually really interesting. So like, yeah, we grow the brains and hit them with, I don't know, it was, I was going to say, like, her name was Nikita. So she was doing all these different flow rates and just popping them over the cells. And like, yeah, actually at accelerated flow rates, you could pretty much modulate activity one way or another. It was really interesting, but she ended up having to go with a different project. So it's unfinished.
Vitaly would love that project because it's all fluid shear stresses, I'm guessing, of it kind of like sucking along the bottom of the glass plate.
Oh my gosh. If you get some new students or like you talk to anybody who's interested in that, like, let me know. I've like an amazing British peristaltic pump that is like, just, it's a great pump. And like, it'd be perfect for those experiments.
Cool. Yeah, I'll tell Vitaly. Jeff, did you have other questions or anybody else?
No, I think I'm okay for now, but I'll definitely, Edmond, I'll get your info from Tyler and probably shoot you over some messages because I'm in the last stretch here of my PhD. And so definitely want things that work and are, success proof. So.
Yeah, sure. I've done the wrong thing so many times. I would be more than happy to share with you the 3 things that I found that worked really well.
Yes, please save me.
Yeah.
Okay.
Definitely. Yeah. Tyler, is that okay with you?
Yeah, of course. I'll be happy to send info over. And thanks a lot, Ed. This was awesome. I really, really appreciate you taking the time.
Yeah, my pleasure. Thanks for the invitation and thanks for hanging out.
For sure. Okay, guys. All right. Have a good night. See ya.
Bye.